today's topic is on protein synthesis. We're going to be talking about how the genetic information on the DNA gets transcribed and then translated into protein. All right, so we're going to start here in the nucleus where the DNA is kept safe. Okay, so here's a picture of a safe, and DNA is a treasure, right? It's kept safely in the nucleus, and your DNA codes for proteins. It is the blueprint to make you, because most of your body, most of the enzymes, the proteins, those are all keeping you alive, and those proteins, the blueprints to make the proteins are kept on the DNA, okay? So on the DNA, you have the blueprints for proteins, and then what's going to happen in DNA protein synthesis is that your cells are going to make an mRNA copy. So RNA is a different type of nucleic acid. mRNA is messenger RNA, which is going to be a copy of DNA, the gene that's going to be transcribed, and this is called transcription. And then from the mRNA will exit the nucleus and go out to the cytoplasm where the protein will be made. And this is done by the ribosomes. If you guys remember, we talked about the type of organelles that make proteins are called ribosomes. So there we have a protein factory as an analogy, and this step is called translation. So the DNA holds the genes, it holds the genetic information to make you. The DNA has the genotype, okay, it has your genotypes, and by transcription and translation, we are making a protein that's going to give you a physical trait, that's going to give you the phenotype, okay? So we're trying to tie everything together that you've learned this year. So here we have genotype and phenotype, which we discussed in the genetics unit, okay? All right, so that protein that's being made is going to give you a physical trait, such as like if you have the gene for the genotype for brown eyes, and you're going to make the mRNA, which is going to be translated to protein that's going to give you the brown eye trait or phenotype. Okay, so let's talk about transcription. Transcription is going from the DNA language to the RNA nucleic acid language. It's going from DNA to the RNA language, okay? Before we talk about mRNA, we do need to talk about RNA. So RNA is a little bit different than DNA. There's 4 main differences. The first main difference is that RNA has a ribose sugar, okay? Not deoxyribose, it has a ribose sugar. It is also a pentose sugar, so it is shaped like a pentagon, okay? Here you have carbon number 1, carbon number 2, carbon number 3, carbon number 4, okay? But it's a pentose sugar because it has 5 carbons, and there's carbon number 5. So if that means then that if we have an RNA strand, this is going to be the 5' end, and then down here will be the 3' end, okay? So the ribose sugar also has 5 carbons. And using the carbon numbers, we know which end is the 5' end and the 3' end. The second difference between DNA and RNA is that you have different nitrogenous bases. In RNA, you have adenine, which pairs with uracil, okay, and then guanine, which pairs with cytosine. So there's no thymine in RNA. You have uracil, which is going to pair with adenine. The third difference between DNA and RNA is that RNA is single-stranded for the most part, okay, not in all cases. For example, I can think of some viruses are double-stranded RNA viruses, but regarding protein synthesis, the mRNA is a single strand, okay? There's also lots of different kinds of RNA. You have mRNA, which is, M stands for messenger RNA. You have tRNA, which stands for transfer RNA. You have rRNA, which is ribosomal RNA. And then you have siRNA, there's miRNA. Those we're going to be talking about when we go over gene regulation, okay, because those are types of RNA that are actually going to silence a gene by destroying the mRNA or inhibiting the ribosome from binding to the mRNA. But that's for another chapter. But it's this is the most recent type of RNA that was discovered, the siRNA and the miRNA. Okay. But anyways, in transcription, in the first step of protein synthesis, we're taking the gene in the DNA and we're transcribing it. Transcribing means to like copy it, but we're copying it in mRNA form. Okay. So that's gonna mean that DNA, the two strands of DNA, are going to split. Okay, they're going to be unzipped, and then one strand of DNA is going to act as a template. Okay, and so remember, with the DNA strand, there are weak hydrogen bonds that hold the nitrogenous bases together. So there's an enzyme, not helicase in this case. It's not helicase. It's another enzyme called Rna Polymerase. And this is the enzyme that is responsible for transcription. Remember, with DNA replication, we talked about helicase, we talked about primase, we talked about DNA polymerase 3 and 1 and ligase. With transcription or protein synthesis, there's only one enzyme that you need to know. It's RNA polymerase, and it does everything. It unzips the DNA molecule, and it also synthesizes the complementary mRNA strand. Okay, so DNA, the two strands will be unzipped by RNA polymerase. One of the strands is going to act as a template. This says template strand, okay? And the untranscribed DNA strand is just called the coding strand. It's the same sequence as the mRNA, except instead of the T's in dRNA and RNA, it's going to be a U, okay? So let me go ahead and just quickly draw. Remember, DNA is anti-parallel, right? So let me just draw my two strands of DNA. It's, oops, I made that. That's supposed to be a 3, the 3' end right there, okay? So this is my DNA strand, and it's going to unzip by RNA polymerase, and then, so let me just draw it unzipped. So which, in what direction will the RNA polymerase be making the mRNA? Just like with DNA polymerase, RNA polymerase can only add to the 3' end, okay? So it's going to be reading the template strand from 3' to 5'. It's going to be reading the 3' to 5' strand as the template, okay? So so this is our 3 to 5' strand down here. That will be our template. And from the template, an mRNA that is complementary to it will be made. This will be the 5' end of our mRNA, and it's going to be made in that direction, 5 to 3'. So just like DNA polymerase 3, RNA polymerase can only add to the 3' end, okay? So, how does RNA polymerase know to bind to the DNA and start unzipping the gene? There's a promoter region, okay? So let's say that this is our gene right here, okay? That's our gene in that dark purple color. Well, right before you hit the gene on the DNA segment, there's a section of the DNA called the promoter. It's called the promoter because it promotes transcription. It's going to promote transcription of that gene. And that promoter has a little sequence at the start of the promoter. It's called a TATA box. I know that sounds really funny. The reason why it's called TATA is because thymine, adenine. Okay, on this strand right over here, there's TA. So the promoter, the TATA box of the promoter is where the transcription factors are going to bind to the promoter. Okay, so you know how I said that the RNA polymerase does everything, right? It unzips the DNA, it makes the mRNA, but it needs help. Okay, even though it's the only enzyme involved here, it needs help, and it needs help from these proteins called transcription factors. Transcription factors are proteins that are going to bind to the TATA box, bind to the promoter, okay, because you're going to see in a minute on the next slide, there's actually more than one transcription factor that's going to bind to the promoter, and it's going to assist the RNA polymerase in binding to the promoter and start to unzip the gene, okay? So there's also, the RNA polymerase also needs help. Besides help from transcription factors, it also needs help from an enhancer region. Okay, enhancer region. It's not shown in here, but you know, on a different chapter, I think the next chapter we'll be talking about enhancer regions. But let's just say upstream from the DNA, so somewhere over here on the DNA strand, you have an enhancer region. It's a segment of DNA that doesn't code for protein. However, it enhances the transcription of the gene, this gene that is downstream, okay? So upstream from the gene, you have the enhancer region. It's just a segment of DNA that's going to help, it's going to help the RNA polymerase bind to the promoter so that this gene can be transcribed. You know, in the last chapter, we talked about how around 97% of our DNA, it doesn't code for protein. 97%. The 97% can be like telomeres. The 97% could be pseudogenes that used to be a gene but now is mutated where it no longer functions as a gene or codes for a protein. There's also part of that 97% of our genome are also enhancer regions. They don't code for a protein. However, they help with the transcription of a gene. Okay? So an enhancer region is a portion of the DNA that's upstream from the gene that's going to help the RNA polymerase bind to the promoter so that transcription can take place. Okay, so RNA polymerase is going to bind to the promoter, and you'll see on the next slide. Okay, so if you take a look at this picture, this is our RNA polymerase, and it's RNA polymerase 2, but you can just call it RNA polymerase. Okay, RNA polymerase is going to bind to that promoter region. The promoter region is this green section of the DNA. Here's our transcription factors. There's several of them shown here. These transcription factors can bind to the TATA box, which is right over here. This is the dark green region. And then other transcription factors are going to bind to the promoter. Enhancer, it's not shown here, but there's an enhancer sequence here that's actually going to fold. Okay, the DNA strand is actually going to fold, and all of those pieces are going to come together and help the RNA polymerase in unzipping the DNA. And you can see that being unzipped right here, and then also adding new RNA nucleotides to the growing mRNA strand. So this is what we call the initiation complex. This is how transcription begins. Initiation complex, think of it this way, the initiation complex is all of these parts coming together so that transcription can take place. And just in a nutshell, what is the initiation complex? You know, it includes enhancer sequence. Okay, it includes the enhancer sequence. It includes transcription factors. And there's more than one usually, okay? Transcription factors, and then the RNA polymerase. All together, they all have to come together. They all have to come together at the promoter region, and you get this initiation complex, and then the RNA polymerase can unzip the DNA at the gene and start making an mRNA transcript that is complementary to the template DNA strand. Okay, and notice in this picture that the mRNA is being made in this, it's pink, from 5' to 3'. And notice how the template strand that is being read, this is the 3' end, so then this is the 3' end, and then this is the 5' end of the template that's being read. Okay, so that's how transcription begins. You need to have all of those things together for RNA polymerase to start unzipping the DNA and then adding complementary RNA nucleotides to the growing mRNA strand. So you can see here, if this is our gene, this is the 3' end, this is the 5' end of the template, this is the 5' end, and then this would be the 3' end of our growing mRNA. So the RNA bases, they match to the DNA template. If this is T, this is G, this is G, then complementary to that is ACC, okay? So let's see if this happens. Okay, yeah, 5 to 3' direction, right? And so the A is going to be complementary to that T. But remember, right over here, instead of a T complementary to the A, it's going to be a U, because in RNA, It's uracil, not thymine. And then that C is complementary to that G. That A is complementary to that T, that G. And then the next one is going to be a U. Oops, sorry, that's supposed to be a U, and an A, and a G, and a C, and an A, and a U, and a G, and a G, and a C, and an A. Okay? So that'll be the 3' end of our mRNA strand. So this would be our gene, the DNA template of the gene, and then this would be the new mRNA that is a transcription, that is a copy of the DNA, but it's a DNA copy in mRNA format. Okay? So after the mRNA is made, for eukaryotic cells such as us, it's actually not that simple, okay? The thing is that when you look at our DNA, okay, when you look at our DNA, there's actually, the gene for a protein is actually cut up into little pieces and then scattered through the DNA, the chromosome, okay? So think of like, think of it, the gene. So let's say this is the gene for like brown eyes, okay? So in our genome, the gene can actually be cut into several pieces, okay? And then they're put in the genome or put on the chromosome in separate places. And then so if they're going to be cut up and put in separate pieces along the chromosome, there's going to be pieces of the genome or pieces of the chromosome that are actually not a part of the gene. And so those segments that are in between the genes are called introns, okay? This is kind of strange, right? Because if you have a gene for a protein, don't you feel like they need to stick together? Because then the mRNA can be just made from it, and then it can go to the cytoplasm where the ribosomes are, and it could be just simply read. But unfortunately, that's not the way that our, actually it is fortunate. We'll talk about a reason why it's fortunate. But so if the gene is split up into several pieces, and it's like, you've got pieces of the gene here. So this is like part, in blue, those are the parts of the brown eye gene, okay, that's been cut up and then put in their genome. And then the non-coding regions are in orange. Those are called introns. Introns are the non-coding regions. Those are like the in-between. So that's the best way to remember that. Introns, in between, those are the ones that doesn't count. That's not part of actually the gene, okay? So your genome has introns. So you know how we said like 97% of our genome doesn't code for anything? Another part of that 97% is the fact that there's introns, okay, introns. Whereas the exons do code for protein. So when we cut up that gene into little pieces, each piece of the gene, the blue here, are actually part of the gene that codes for the protein, the brown-eyed protein, okay? We call those exons, and they're called exons because EX mean those are the parts of the DNA that are actually part of the gene that is going to be expressed, okay? Expressed meaning like transcription, translation will take place, you'll get a protein, okay? Whereas introns are in between. So introns are in between, whereas exons are part of the gene. Those are the ones that are going to be expressed. So then you know how we had the promoter. So let's pretend now that we have the promoter here. So here we have the promoter. The RNA polymerase binds to the promoter. It unzips the DNA. It starts like making an mRNA that is complementary to the gene, we said, to the template. Well, that template has introns in there. It has introns in there. And so the mRNA is going to have introns as well, okay? So the mRNA is going to have introns as well. And we do not want those introns to be part of the mRNA when it goes out to the ribosome. Because when the mRNA goes out to the ribosome just reads the mRNA and starts, making the protein from it. So we want to remove the introns before the mRNA leaves or exits the nucleus, okay? So the introns have to come out. They have to come out. And that's what we call splicing. Like splicing sounds sort of like slicing, cutting. You're cutting out the introns. And so this is called post-transcriptional processing, because after transcription, what do you get? You get an mRNA strand. So do you guys see right here? This is the DNA with the introns, which are in orange. Those are the introns. And then also the mRNA has those introns. It's the mRNA copy of those introns. And so what your cells do is it removes those introns. So these little orange segments are the introns. They get cut out, or they get spliced out. That's how we say it. They get spliced out, and then the exons are pieced together, and you get a mature mRNA transcript. I don't know if you guys can read it because it's so far below. It says mature mRNA transcript. Okay, transcript. And so, eukaryotic DNA may have like 10,000 nucleotide bases in that gene. Okay, in that gene. But that gene contains non-coding and coding segments. And so the mRNA is going to have 10,000 RNA bases. The pre-mRNA is going to be about 10,000 bases long, but then the introns are going to be spliced out, and you're left with about 1,000 nucleotides that actually code for the protein. So the mRNA splicing means that we're editing out, cutting out the introns, and we're piecing together the actual pieces of mRNA transcript that codes for the protein, okay? So the reason why we have mRNA splicing is because your genome actually has a lot of introns that are in between the coding regions of the gene, okay? And you know, the two scientists that helped discover introns and exons, they were actually studying the common cold. Just like with eukaryotic genomes, the common cold actually has introns and exons as well. So when they were studying the common cold, they noticed that the mRNA contained these introns and exons, and then they were being removed. Okay. So splicing must be accurate. There cannot be any room for mistakes, because we're going to be talking about translation in a few minutes. Translation is where the ribosome is going to be reading the mRNA in a reading frame. Okay, reading frame. And the reading frame is 3 nucleotides of the mRNA called codons. Okay, so like this is, it's kind of hard to see with the colors here, but this is a codon. AUG is a codon. CGG is a codon. CTA is a codon. TGG is a codon. GUC always reminds me of guac, like guacamole. CGA, those are all codons, okay? And the ribosome reads each codon, and each codon codes for a specific amino acid. That's why they're called codons, because they code for UAA is a codon, and it codes for a very specific amino acid in all living things. It's amazing how in a bacteria, UAA codes for the same amino acid as UAA in my cells. Okay, and that's exactly how, you know, in biotechnology, we can have bacteria make insulin, human insulin for us. All we need to do is put in the human gene into their genome, and then now they have our genes in them, and then when they make the mRNA for insulin, then their ribosomes recognize the codon, and they can read the mRNA codons and make the human protein for us, because the codons are read exactly the same. UAA codes for the exact same amino acid in a bacteria as in my cells. Okay, so UAG actually, it says right here, it codes for a methionine amino acid, or MET. Okay, CGG codes for the amino acid arginine. You know, this is actually, I know why it says the T's. I was like, T's, there's no T's in RNA. It's because this is showing you if there was a mistake. This is going to be where this part of the DNA is deleted. Okay. So and then the mRNA that's going to be read, UCC is going to code for serine. GUA is going to code for aspartic acid or asparagine, etc. Okay? But let's say that there was a mutation. where this segment is deleted. It's now gone. Let me just make sure that CTG. Okay, so now it's going to be, yeah, it's right over here. So now this section is missing. Okay, now it's deleted. And so this is the reading frame. So AUG. So originally it was AUG, CGG, UCC, GAU, AAG, GGC, CAU. Those are 1, 2, 3, 4, 5, 6, 7. Those are 7 codons for 7 amino acids. But if we delete this segment of DNA, then how many codons do we end up with? 1, 2, 3, 4, 5, 6, 7, and then this is just continuing on beyond this point. Okay, so AUG is the next reading frame, which is the original codon, which codes for methionine, and then CGG codes for arginine. But remember how we said that this segment of the DNA is deleted? Then where it's deleted, everything moves over one place. So see this G right here? This G and everything after it shifts over. It's going to shift over, and then now you're going to end up with the reading frame over here where it's going to be VAL. It's going to code for valine instead of serine. Okay? So here's the CGG. Here's that G. Wait, which? Oh yeah, it's deleted right there. So here's that G, okay? So CGG and then GUC instead of UCC. So UCC codes for serine, where now it's GUC, which codes for valine, okay? And then now we have CGA. Everything just moves over, and as a result, all of the amino acids following that deletion is completely changed. It's completely changed. Instead of serine, asparagine, lysine, glycine, histidine, it's now valine, arginine, and then UAA is actually a stop codon, which means that the protein translation synthesis stops right here, and then this is the amino acid sequence. It's a very short protein, a very short amino acid sequence. It's supposed to be 7 amino acids long, right? But instead it ends up being 4 amino acids long. Okay, but more on that in just a minute. I think I kind of got ahead of myself there. I was already discussing translation. But with the splicing, how I said splicing occurs where the introns come out and the exons stay together. There are enzymes that do this, okay, in the nucleus. These enzymes are called spliceosomes, okay, spliceosomes. And the spliceosome is made up of SNRPs, not Smurfs. I think you guys might be too young to know the Smurfs, okay? But when I was growing up, Smurfs was on TV, the Village of Blue People. And spliceosomes are made up of SNRPs. And what are SNRPs? SNRPs are made up of RNA and protein. And the RNA and protein, when they get together, we call it, spliceosomes, which are going to cut out the intron. So here you can see this is the intron. The intron gets folded, and then the exons come together. You can see that here, come together, and then the spliceosomes cut the intron out, and then they're going to glue the exons together, and then the intron is removed. What happens to the intron? Remember, this is in the nucleus. So those intron nucleotides, that RNA nucleotide is just going to get cut up and then reused. It can be reused to make more mRNA. And then Fillmore says, "Whoa, I think we just broke a biological rule." Okay, the reason why he says that is because you know how we said enzymes are proteins. Spliceosomes are enzymes, but they're not 100% protein. Spliceosomes are enzymes, but they contain RNA. Okay, they contain small nuclear RNA, which is not a protein. It's RNA nucleotides, but it acts as an enzyme. Okay, so the biological rule that we're breaking here that Fillmore is talking about is that we say, enzymes are proteins. Well, in this case, this is an example of where the enzyme is not completely a protein. Part of the enzyme, the spliceosome, is actually RNA. Okay, so this is actually really cool, alternative splicing, and there's a lot of research going on right now about, you know how we talk about genome? There's a lot of research now about proteome. Oops, I meant to write a P. I wrote a G. Proteome. So, you know, what do they study when they study proteomes? They're studying, the genome is basically all the genetic information in a person. Proteome studies, they study all the proteins in an organism. Okay? And the reason why I bring that up is because, you know, they say that we have about 20,000 genes in the human genome. I'm going to write this down just so that you guys can see visually what I'm talking about. We have about, you know, 20,000 genes. But we have, and you guys, a number of proteins, it varies depending on what like website you check. But I'm going to go with the higher end. Okay, I'm going to go with the higher end. There's around 400,000 different proteins in our body. And you know, today in the video, the guy said there was like over 2 million proteins. And I'm like, maybe 2 million proteins in a cell at one time, but we have 400,000 different proteins. So how is that possible? You know how we said, oh yeah, one gene makes one protein. That doesn't make sense, because if we have 20,000 genes, that means that we would have 20,000 proteins, right? Well, this is where alternative splicing comes into play. Basically, what's happening is, you know how I said genes, you have these introns in between the cut up genes? Sometimes those exons can be introns, and sometimes those introns can be exons. So from one gene, which is cut up into introns and exons, you can actually get more than one protein. Okay, so 20,000 genes, you're going to end up with around 400,000 proteins, because one gene actually can code for more than one protein. And this is how it's done. Okay, so here we have our DNA sequence, and let's say these are the exons. You have blue, green, brown. I guess that's brown, or is that orange? And then blue exons. And then in transcription, so let's say transcription is taking place, RNA polymerase is going to unzip the DNA, make the mRNA transcript with the help of the transcription factors and the enhancer sequence, right? So transcription is going to happen, but then when splicing occurs, so this is considered to be an intron. This is considered to be an intron. This is an intron. This is an intron. You know, you can have the exons. So I'm going to just write down 1, 2, 3, 4, 5. So we have, you know, the 5 exons. 1, 2, 3, 4, 5. So those 5 exons make up a gene. However, those 5 exons can make one protein by splicing out this one. Okay, so this exon can be treated as an intron, and as a result, you're going to get 1, 2. You're going to get 1, 2, 3, 5. So 1, 2. I don't know if you guys can see it in the video because my full screen doesn't necessarily capture everything. You're going to end up with an mRNA strand that has segments 1, 2, 3, 5, missing 4. Okay, or in this case, exon 3 is considered to be an intron for a different protein, and exon 4 is considered to be an exon. Okay, so you're going to end up with segment 1, 2, 4, and 5. So you end up with 2 different mRNAs. You guys see here? You end up with 2 different mRNAs. That's called alternative splicing. So from one gene, which has 5 exons, you can actually get more than one protein, depending on which exon is going to be treated as an exon, or which exon is going to be treated as an intron. Okay, so do you guys see here? I can end up with 2 different kinds of mRNA, which is going to give me 2 different kinds of proteins. Related proteins, because there's segments of the mRNA that are similar. They're related proteins, but they're different because they're going to have different codons. Okay, so that's called alternative splicing. That explains how We have 20,000 genes, but we can get so many more proteins because of alternative splicing. So yeah, it's starting to get hard to define a gene, because I said that this is a gene with exons 1, 2, 3, 4, 5, but depending on the protein that the cell is trying to make, some of these exons may actually not be an exon. Like this one would be this one could be considered an intron to make this the first mRNA example here. So after the mRNA is spliced out, okay, after the mRNA is spliced, then there's something else that occurs. Right before the mRNA is going to be moved to the cytoplasm, the mRNA needs to be protected, because in the cytoplasm, there are a lot of enzymes called nucleases, and these nucleases are going, let me just write it down, nucleases. And these nucleases are in the cytoplasm, and they basically digest, break down RNA, or nucleic acids, I should say. Okay? So your cells want to protect that mRNA because the mRNA needs to get to the ribosome so that the protein can be made. So what's added to the mRNA is a 5' cap at the start, at the 5' end. It's called a 5' cap. There's a special GTP, guanine triphosphate, a very special guanine triphosphate that's added to the 5' end of our mRNA. And then there's a poly-A tail that's going to be added to the end. Poly-A means those adenines. Okay? So a bunch of adenine RNA nucleotides are added to the end. It's called a poly-A tail. So, you know, if those nucleases come along and get to the mRNA, it can start it can start digesting the poly-A tail, and the mRNA will have some time to remain intact so that the ribosomes can go ahead and start reading the mRNA and making the protein. And then the mRNA, or the 5' cap, cannot be broken down by the nucleases because of its structure. It's not recognized by the nucleases. Okay, so I'm actually going to pause my recording here, and then I'll continue recording probably tomorrow, okay? All right. Okay, so it's a new day for me, and we're going to be talking about translation today. And translation is right after transcription, where after the mRNA is made, then the mRNA is going to go to the cytoplasm, where the ribosomes are going to read the mRNA and make the protein. And as you guys know, the building blocks of the proteins are the amino acids. Okay? All right, so here we have the DNA. The DNA has the genes, right? So the DNA, remember, DNA has T's and C's and G's and A's. So if this is the DNA sequence, there are 4 different kinds of nucleotides that make up the DNA. And then there are also 4 different kinds of nucleotides that make up the mRNA. This is going to be complementary. The mRNA is complementary to the DNA template. So if this is the 5' end of our mRNA and this is the 3' end of our mRNA, then this means that the template, this is the 3' end and this is the 5' end. Okay? And every three RNA, mRNA nucleotides is what we call a codon, a codon. You guys have heard of the genetic code before. What does a code mean, or codon? The genetic code refers to the codon. That codon, every 3 on the mRNA, it codes for a specific amino acid. AUG codes for the amino acid methionine. CGU, it codes for a specific amino acid called arginine. And then GUA is valine, etc. Okay, so all of these amino acids, ASN, ALA, CYS, ALA, those are all the 3-letter abbreviations for amino acids, and you don't need to know the names, specific names of the amino acids. Just know that the codons, every 3 on the mRNA, codes for a specific amino acid. And we're going to be going over the genetic code chart very soon. You're going to need to know how to read that chart. You're going to need to know how to find the codon on the chart and then figure out which amino acid that codon specifically codes for. Okay, so this is basically the same slide we just went over. If this is the DNA template, this is the 3' end, this is the 5' end. Then this is the 5' end of our mRNA strand, and then this is the 3'. I'm like super delayed with the video and the sound is completely off, but it's okay. Okay, so Francis Crick, Watson's partner, I mean, he had determined that there was a 3-letter triplet codon system, but the 2 scientists that proved this was Nierenberg and Codons. So what they solved was that every 3 letters in the mRNA codes for a specific amino acid. It's sort of like in this sentence right here, every 3 letters is a word. On the mRNA, every 3 letters it represents or it codes for a specific amino acid. And the very first codon that they discovered was UUU. They determined that UUU, it codes for an amino acid, phenylalanine, okay, or PHE, okay? So here's a picture of Nierenberg and Codons. Nierenberg was an American geneticist, and Codons was an Indian, so he's from Asia, India. He helped together, they worked together and they helped discover that the codon, UUU, codes for phenylalanine amino acid, and they won the Nobel Prize for their discovery back in 1968. So here is the genetic code chart. And the really cool thing about the genetic code is that it is a code for all life. When you look at a bacteria, when you look at human cells, when you look at plant cells, every codon codes for the exact same amino acid. So the start codon, AUG, it codes for methionine in a bacteria, in a human cell, in a rat cell, in a little ant cell, okay? AUG, mRNA codon, it's going to code for the same amino acid. So how do we use this chart? Well, if the first letter of the codon is A, you're going to look to this, the left side of the chart. The first base, the first nitrogenous base on the 5' end, A is our first letter. So that means that the amino acid that we're looking for is somewhere, okay, it's somewhere in this row. So I'm going to go ahead and like circle this entire row. Okay, and then the second base of our codon is U. And so that means when you look at UCAG at the top, that means that our amino acid is somewhere in this column. And we already established that the amino acid is somewhere in this row. That means that the amino acid that we're looking for is somewhere in this box. Whereas G, which is the third letter of the codon, and I need to move my head here. Let me move it out of the way. Okay. Whereas the third letter of the codon, so you're looking at the far right-hand side of the chart, you're looking at UCAG. We're going for G. AUG was that codon. G was the last letter of the codon. Then you're going to, it's somewhere in this row, which means that AUG, it codes for MET. MET. MET is methionine. It is the starting amino acid for every protein, okay? Because AUG is always the start codon on an mRNA molecule. It is the start of the mRNA molecule. It is the start of the chain of amino acids that it codes for. It starts with MET, methionine, okay? Some other thing about the genetic code that you might have noticed that, for example, leucine or LEU, there are 6, okay? There are 6 codons that code for the same amino acid LEU. So that's what we mean by when we say the code is redundant, because there are several codons, except for AUG. That's the only codon that codes for methionine. All the other amino acids, they're multiple. More than one codon that codes for it. So that's what we mean by saying redundant. And when you look at, for example, this group of 4 codons that code for leucine, they're all the same. The first and second letter of the codons are the same. It's the third letter of the codon where we consider it like the wobble. Okay, that's the one that can be changed. That's the one that can be different. And that's good because if there was a mutation and it happened to be in the third letter of our codon, then that's okay because it's going to still give us, the same amino acid. And if it gives us the same amino acid, it's going to give us the same protein. The protein isn't changed or altered in any way. So that's a good thing. That's one of the good things about the redundancy of the genetic code. So AUG, we said, is the start codon. And then you may have seen there are 3 stop codons, UAA, UAG, UGA. Those are the 3 stop codons. There are no amino acids that are coded for by these codons. Instead, it's a sign. It's a symbol for a release factor to enter the ribosome in translation, which we'll be talking about in just a minute. It's a sign for the ribosome to stop translation. The protein is now made. The protein is now completed. It's finished being made. Okay? Oops, let me go ahead and lower the volume. Didn't realize it was that loud. Okay, so those are AUG is a start codon, and then UAA, UGA, UAG are the stop codons. Okay, and Fillmore is asking, why is the wobble good? And I mentioned this before. If that third letter of our codon is different, if there's a mutation, it's okay because the amino acid isn't changed. If the amino acid doesn't change, therefore the protein shape is not going to be changed. The protein will not be altered in any way. Okay? So how are codons matched to amino acids? Looking at that chart, AUG, it codes for methionine. CGU, it codes for arginine. GUA codes for valine. Like how is the cell going to bring those amino acids and bring them together to make the protein? Well, there's a tRNA involved and there's a ribosome involved. We haven't really talked about ribosomes or tRNA yet, but we'll do so now. Okay, the tRNA is going to be attached to the appropriate amino acid, bringing the amino acid to the ribosome. So AUG on the mRNA is going to match up to the tRNA anticodon. Okay, so there's something called the anticodon. Anticodon just means opposite to the codon. So if the codon is AUG, on the tRNA molecule on it, there is a UAC anticodon. They are complementary. And in fact, they are going to form temporary hydrogen bonds with each other, holding that methionine amino acid in place inside the ribosome. Okay. What I'm saying right now might be a little confusing because there's no drawing of a ribosome, but I'll be drawing a ribosome and discussing translation in further detail in a few minutes, okay? But what I want to say is that CGU is the next codon. So the next codon codes for the amino acid arginine, which is brought to the mRNA codon using a tRNA, and the tRNA anticodon is GCA. So on the tRNA, you have these anticodons that match up, that are complementary to the codon. And those tRNAs are bringing the appropriate amino acid, okay? So that genetic code chart, that you need to remember that the genetic code chart, that chart is referring to the codons on the mRNA, not the anticodon. Because if you look at the codon chart, CGU, it's going to code for arginine, but GCA, which is the anticodon, it codes for something else, okay? So for example, let me actually go back. GCA, okay, G, and then C, and then A, that means that the amino acid that we're looking at is alanine, okay, alanine. And that is not, GCA is the anticodon. That is not the amino acid that corresponds to that codon. The codon is CGU, anticodon is GCA. So that genetic code chart is only referring to these codons, not the anticodon, okay? So you cannot use the anticodon to determine the amino acid. You have to look at the codon to determine the amino acid that's being brought to the ribosome. So this is what tRNA looks like. Again, it is a type of RNA. RNAs have the A's and the U's and the G's and the C's. Those are the four nucleotides that make up RNA. And just like DNA, it has a 5-carbon sugar, which we already talked about. So there is a 3' end and a 5' end. Okay, so this is a strand of a strand of RNA, a single strand of RNA that happens to be folded in a few areas. Okay, and the folding is actually due to hydrogen bonding that forms between complementary base pairs on the single strand. Do you guys see that those little dots are holding this tRNA in a specific 3-leaf clover shape, this 3-leaf clover shape? So C goes with G down here, U goes with A, C goes with G. So there is some hydrogen bonding that's keeping the tRNA in shape, in this 3-leaf clover shape, okay? So the 3' end, though, is where the amino acid is going to be held. So the amino acid is brought to the ribosome. It's attached to the 3' end of our tRNA. The middle leaf right here is where the anticodon is found. Okay, that's where the anticodon is found. So if the anticodon is AAG, then the codon is going to be UUC. Okay. Well, that will be the 5' and then that's the 3'. So UUC. Okay. This is another type of drawing of tRNA. I kind of don't like this one because it just visually it's not really easy to understand, but it's the same thing. It's just sort of a 3-dimensional structure model of tRNA where it's like highly folded, and then you have the hydrogen bonding that happens between complementary base pairing, and then these are the anticodons, which look kind of crazy, okay? And then the amino acid attachment is at the 3' end. Here's another version, a drawing of tRNA. So this slide shows you 3 ways that textbooks actually show the drawing of tRNA. This cylindrical representation of tRNA is very common, and actually it's used in our textbook. So keep in mind that even though it's simplified as a cylinder, it's actually a 3-leaf clover-like shaped structure, okay? And then the anticodon is down there. So if the anticodon is AAG, the codon is UUC that's the codon on the mRNA, okay? The anticodon. Okay, and so you know the tRNA is bringing the amino acid to the ribosome. How did that tRNA even get the amino acid? So I'm just going to draw my 3-leaf, that's my 3-leaf clover. This is the 3' end, this is the 5' end. And then there's, that's my anticodon there. There's 3 nucleotides that act as the anticodon. The 3' end carries the amino acid, and the amino acid is attached to the 3' end using an enzyme called Aminoacyl tRNA synthetase, okay? You don't need to memorize the name of that, but it's just an enzyme that brings the amino acid and the tRNA together so that that tRNA can take that amino acid to the ribosome, to the matching mRNA codon. So if this is our codon UGG on the mRNA, so this is our codon, right, on the mRNA, the anticodon on the tRNA is opposite to the codon. It's going to be ACC. But when we look at the genetic code chart, UGG, the codon, it codes for tryptophan, okay, tryptophan or TRP. Okay, before we talk about translation in detail, we do need to talk about ribosomes. So we've learned in previous chapters that ribosomes are the organelles that make protein. They are the protein factories, right? This is where the ribosomes are where the tRNAs and the mRNAs meet. The mRNA has the codons, the tRNAs bring the amino acids to the mRNA, but the meeting place would be the ribosome, okay? And the ribosome has 2 subunits. There's a large subunit, okay? And then there's a small subunit. In this diagram, they kind of look like boxing gloves. I like to draw, like I prefer like this drawing of ribosomes where it's like a hamburger bun, okay? A top bun, which is nice and tall, and then a small bottom, skinnier, flatter hamburger bun. And those two subunits come together. They come together to read the mRNA during protein synthesis or during translation, okay? And this large subunit actually has 3 pockets. There's 3 pockets where the tRNA can go in. There's 3 pockets. And the first pocket is called the E site. This middle pocket is called the P site. And then the far right side, that pocket where the tRNA can go in is called the A site. So the 3 sites are called EPA. EPA. Those are the 3 sites or pockets in the large subunit of the RNA or ribosome. Okay? So yeah, those are the 3 sites that I was talking about, EPA. And you can see in this drawing, a tRNA has brought the very first amino acid into the P site. This is a picture. This is actually a good picture of the initiation complex. This is called the initiation complex. This is what starts off translation. Initiation complex. You have the tRNA bringing the first amino acid, met or methionine, to the mRNA, which is, actually the mRNA is sandwiched in between the 2 hamburger buns, the 2 ribosomal subunits, the large subunit and the small. So yeah, that you can think of the mRNA as being like a really long burger, okay, burger patty, and then the ribosome like the hamburger buns. It's like the mRNA is just sandwiched in between, but then the burger bun, the hamburger bun, has 3 slots or pockets that the tRNA can go in and fit. Okay? Notice the anticodon is opposite to the codon. It's found on the tRNA. Okay? So this is what we call the initiation complex. So there are 3 steps to protein synthesis, or actually I should say not protein synthesis, I should say translation. Okay, there are 3 steps to translation. Well, I don't know why the video and sound and everything is very laggy today. So initiation is the start of translation. This is, as I just mentioned previously, this is where the mRNA, the ribosome, and then the tRNA bringing the first amino acid met to the ribosome. Okay, so those are the things that need to come together to initiate translation. And then elongation is when the next tRNA, so you guys see this tRNA, is being brought into the A site. It goes into the A site. And then once the 2 amino acids are sitting side by side in the ribosome, they're going to form a peptide bond. Okay, this is called elongation, when the amino acids come in, and then elongation, the peptide bond forms. Once a peptide bond forms between the first and second amino acids, the first tRNA will release met, leave, it's going to exit. Okay, it's going to exit the ribosome through the E site. Do you guys see that? It's now in the E site and it's going to leave. And then the second tRNA is now in the P site, holding on to met and leucine. And then a third tRNA is going to come in, and apparently I guess the third, oh, I can't really see, but AAU, I'm not sure what that codes for, but AAU codes for a specific amino acid carried by a tRNA into the A site. Then they'll form a peptide bond. Once that peptide bond forms, then this tRNA is going to release leucine, okay, and then it's going to move into the E site. Okay, and then here UAA is a stop codon, then a release factor will go into the A site, and then that's a sign to the ribosome that the protein synthesis, or I should say translation, is now finished. It's now done. Okay, so those are the 3 steps for translation: initiation, elongation, and termination. You know, I'm going to go ahead and like pull up a whiteboard and kind of draw the whole steps of translation. All right, so hopefully this will work out okay. So we're going to have our mRNA, So this is the 5' end of our mRNA. This is the 3' end. Let's just do, AUG is the start codon. Okay, AUG, CGG. I'm just making this up as I go. Oh no, I can't do that. Well, I can. No, because that's going to end up being a stop codon. So let me go ahead and erase that last A. Because that's going to... Oh wait, no, the stop codon is the UU, so never mind. So I can't do AA. So we'll do A, C, A, C, G, C, A, and then U, A, right? Okay. So this is the start codon. And then this is CGG, which is the second codon, and this is the third codon, all the way down to the stop codon. Okay, so the initiation complex is when a big ribosome, okay, the 2 subunits come in, and like I like to draw it like this, right? Because I said that it's like a hamburger bun. It sandwiches the mRNA. The AUG first codon, that's the first codon. This is the E site, this is the P site, and this is the A site of our ribosome. Let me use a different color because I'm going to use a different color for the tRNA that's going to bring the amino acid. Maybe I'll use blue. Okay, so the first amino acid, and I'm going to draw a clover shape, brings the amino acid MET. Oh, I hope that you guys can see this because whenever I do screen record, it always chops off the top and the bottom. But this is the tRNA that brings the methionine or MET amino acid to the P site. Okay, also what I'm going to draw on my 3-leaf clover tRNA is the anticodon. If the codon is AUG on the mRNA, the anticodon is going to be UAC. Okay, so this is the initiation complex. This is the first step of translation. The next step is elongation. So the next amino acid is going to come along and go into the A site, and it's going to bring the next amino acid, CGG. I don't know what that codes for. I'm just going to write AA for amino acid. So now, oh, and I should write down the anticodon. So on the tRNA, the anticodon then will be GCC, right? So now that these 2 amino acids are sitting side by side, they're going to form a peptide bond. Okay, they're going to form a bond, a peptide bond. Once a peptide bond forms, this first tRNA is going to release methionine and allow for that methionine to be attached to the second amino acid. Okay? And then next what's going to happen is that this ribosome is going to move over, or actually the mRNA is going to shift over. So should I draw this again? Or maybe I can just erase the ribosome. I'm going to grab my eraser tool. Oh, nice. That was pretty easy. Okay. Okay, no, that was pretty easy. That's good. All right, and then, so I'm going to redraw the ribosome. The ribosome will now be shifted over. I'm going to draw, maybe I should draw the, no, I'll continue just to draw it in red. So now the ribosome has moved over like this. There's my hamburger buns. The first tRNA is now in the E site. So this is the E site. This is the P site. This is the A site. The A site is empty, okay? Now that this tRNA is in the E site, it's going to exit. It's going to leave the ribosome, and it's going to leave behind methionine attached to the second amino acid. So I'm going to try to erase. There we go. Okay, so it left behind MET. All right, and then now the next amino acid. Let me do blue. The next amino acid is going to be brought by the second tRNA into the A site. And again, I don't know what AAC codes for because I don't have the table in front of me. So I'm just going to write AA, short for amino acid. And the tRNA anticodon is UUG. What's going to happen between those 2 amino acids? Hopefully you know the answer to that. They're going to form a peptide bond. they form a peptide bond. So now we're getting this chain of amino acids. That is what a protein is, right? All right, after the peptide bond forms, then this tRNA is going to leave behind that growing chain of amino acids. So I'm going to go ahead and erase that. Okay, erase this tRNA. And actually, I'm going to erase the ribosome, because now the ribosome is going to be shifted over. And this tRNA specifically will now, this last tRNA will now be in the P site. Oh, I gotta erase the ribosome. Okay, so now I'm gonna redraw my ribosome in red. Okay, so yeah, it's gonna be my big ribosome. And then this is the E site, this is the P site, this is the A site. The A site is empty. It is ready for the next amino acid, right? And the next amino acid is going to be brought by the tRNA. Okay, so the next tRNA, and if I look at the, the genetic code chart, CAC. What does CAC code for? I don't know, but it's going to bring that specific amino acid. And the tRNA anticodon is going to be GUG. What's going to happen between these two amino acids that are now side by side in the ribosome? They're going to form a peptide bond. That bond is very long compared to the other ones, but it's just imagine that they're all like equally distanced, okay? So you have a peptide bond that forms between them. And you guys, this is just going to continue on until it reaches a stop codon, but let's just continue going for, the sake of this drawing. Okay, so I'm just going to erase that. And then, actually, you know what I'll just do is I'm just going to draw it as if we just reached the end, okay? I'm going to go ahead and draw my ribosome, okay? So then what's going to happen is you have, oh, I was going to draw it like this, yes. And then I need to draw the next tRNA that brought the next amino acid. Okay, so there's my next amino acid. They formed a peptide bond, right? And then this tRNA in the the E site is now gone. Okay, this guy is gone. All right, so we have here a chain of amino acids, and it looks like 5 amino acids, right? And once the ribosome reaches that stop codon, that stop codon is, let me just get my pen color. I should just go back to, I'll just keep it blue. Okay. So that stop codon is, there's no amino acid that codes for that stop codon. So a release factor, it's like a protein that's going to go in. Maybe I'll just draw it like the way that your book has it, RF for release factor. Okay. The protein goes in, and then it's a symbol assigned to the tRNA and the ribosomes and the mRNA to just come undone, like they all come apart, and now you have this chain of amino acids, which we consider to be a polypeptide, right? That polypeptide is called a polypeptide because there are many amino acids joined together by peptide bonds, and that's going to make a protein. Okay, so that's translation. You have a ribosome. You have the mRNA in between the large and small subunits of the ribosome. And then you have the EPA sites of the ribosomes where the tRNAs are bringing the amino acids to the ribosome. Okay? So that's initiation, elongation, and termination. Those are the 3 steps of translation. Okay? Let me see if I skipped a... No. Okay. So, that amino acid, that mRNA, like, how does it know where to go, okay? There is a signal protein that is, actually there's a signal protein on the growing chain of amino acids, so not mRNA. See this right over here? This is the chain of amino acids that are starting to come out of the ribosome, there's a signal recognition particle that attaches to it, and then it's going to cause the ribosome, which is still reading the mRNA and building that chain of amino acids, to go where it needs to go. So if this is like for example, this is the rough ER, you know, that signal is telling the ribosome that the protein that's being made is going to either be packaged in a vesicle, so that maybe this protein is an enzyme. It's going to be maybe a digestive enzyme that's going to be part of inside a vesicle, which is going to become a lysosome. Do you guys remember lysosomes? Lysosomes are little vesicles that contain digestive enzymes. So maybe this protein is an enzyme that's going to digest. So then it'll become, you know, it's going to pinch off of the ER, and then you're going to have that protein in there, and that's the enzyme. And this is now called a lysosome. And that signal protein, a signal recognition particle protein, it can, you know, tell that growing polypeptide chain to stay in the cytoplasm. The ribosome is going to stay in the cytoplasm. Maybe it's an enzyme that's going to play a role in cell signaling or something, or a cascade, a kinase in the cytoplasm. Okay? So the signal peptides, they're just basically proteins that are going to tell the growing amino acid chain where it needs to go, because the protein is either going to be secreted out or maybe stay inside. Okay? All right, so let's go ahead and just summarize protein synthesis, transcription, and translation, right? Those are the two steps of protein synthesis, transcription. And remember, in transcription, we're going from DNA to mRNA. The mRNA is being made. Oops, that's supposed to be M, okay? mRNA is the product of transcription. And then the second step of protein synthesis is translation. This is where a polypeptide chain is being made, okay? A polypeptide chain is made, okay? All right, so let's start with step one. We start with the gene in the nucleus. What enzyme is this, you guys? This is RNA polymerase. RNA polymerase is the enzyme that's going to bind to the promoter, okay? But you guys remember, the RNA polymerase needs help. What does it need help from? It needs help from two things. It needs help from transcription factors, number one. And then the second thing it needs help from is the enhancer sequence, okay? So there's an enhancer sequence somewhere upstream here, okay, where it allows for the RNA polymerase to bind to the promoter. So in this picture, I only see the RNA polymerase, but there's actually transcription factors that are promoting transcription. So RNA polymerase is going to unzip the gene, unzip the DNA, and then start making a complementary mRNA sequence. This is the 5' end. This is the 3' end. It's adding to the 3' end. And then once transcription is finished, then what's the second step? Okay. This is our pre-mRNA. Pre-mRNA, remember, is before the mRNA is finalized and exits the nucleus to the cytoplasm. So what's going on here? The introns are going to be cut out. Splicing, okay? And then also after the introns are cut out, or splicing takes place, the 5' cap is added. So this is our 5' cap, and then this is our poly A tail, right? Now it's a mature mRNA that can safely exit the nucleus and go out to the nucleus, where translation will take place, out to the cytoplasm, to where the ribosomes are. You have the initiation complex, which is the ribosome and the mRNA and the first tRNA bringing the first amino acid methionine to AUG. So AUG is the start codon, maybe somewhere over here, right? Okay, oh, I forgot to mention here, so after the tRNAs drop off the amino acids, they actually go, out to the cytoplasm, and then they bind to the amino acids that are floating in the cytoplasm, and they'll just come back to the ribosome. This is the enzyme, remember the enzyme that brings the amino acid to the tRNA and brings them together, okay? So that's what's going on in this picture. Once the tRNA is done dropping off the amino acids, they'll they'll pick up another amino acid using that enzyme. All right, and then that ribosome is just going to continue reading the mRNA strand until it reaches a stop codon, okay? Once it reaches the stop codon, that means that the protein polypeptide chain is done, it's being made, it's finished, and then they'll disassemble. The ribosome, 2 subunits will disassemble, release the mRNA, okay? So that is, the whole story of protein synthesis. And now we're going to talk about other stuff, okay? So in prokaryotes, they don't have a nucleus, right? They don't have a nucleus, so transcription and translation will happen at the same, they're going to happen at the same time in prokaryotes. I don't know why it doesn't say translation here, but transcription, translation for prokaryotic cells happens exactly at the same time, and I'll show you a picture of that coming up in just a minute. But let's go ahead and compare between prokaryotic and eukaryotic genes. In prokaryotic cells, the DNA is not in the nucleus, it's just in the cytoplasm because there is no nucleus in prokaryotes. Their chromosomes are circular. There is one circular piece of chromosome, whereas in eukaryotic cells, you have linear pieces of chromosome. For example, humans have 46 linear chromosomes. Their DNA is naked, meaning there are no histone proteins that the DNA is around. There's no introns in prokaryotic genome, whereas in eukaryotic genomes there are introns and exons, okay? This is one of the reasons why prokaryotes, like bacteria, they evolve so quickly. This is one of the reasons why they evolve so quickly, is because they have no introns. So if there's a mutation, it's going to be a mutation in a gene. And so they're going to be changing in that way, okay? They're going to evolve in that way because there's going to be more mutations that are actually going to cause changes. Whereas because, earlier on in the video, I did say 97% of our genome doesn't code for protein, but there are, it depends on what source that you're looking at. It's 97 to 99%, okay? So between 97 to 99% of our genome is introns, or doesn't code for actual protein. So if there's a mutation, the likelihood of that mutation affecting us as phenotypically is very small, okay? Because only 1 to 3% of our genome is actually considered to be part of a gene, okay? And you guys remember what introns and exons are, right? So as I was saying earlier, that transcription and translation happens at the same time in prokaryotes like bacteria. You can see, let me just describe what's going on in this microscopic picture. This is the DNA, okay, that's the DNA strand. This is a drawing version of that microscopic picture. This is the DNA, and this is the RNA polymerase. And so the RNA, let me just draw another RNA polymerase. So this is RNA polymerase making the mRNA. So there's our mRNA that is complementary to that DNA strand. And then what's going on in this picture is that those are ribosomes. Okay, those purple things are ribosomes. They immediately latch on to that mRNA. I should have drawn like the big hamburger bun and the small hamburger bun, okay? They attach to that mRNA and just start translating, start making the protein. So they just they grab on to the mRNA and they just start making the protein. And this is what we call polyribosomes, because there are many ribosomes on that mRNA making a lot of proteins. Each ribosome, there's like 20 ribosomes on this mRNA, and each one is making a protein or a polypeptide chain. So with prokaryotic cells, they can make a lot of proteins in a short amount of time because transcription and translations happens at the same time, okay? So again, this is just showing like in a bacterial cell, transcription translation happens exactly at the same time. Whereas with us or eukaryotic cells, transcription happens in the nucleus, then the mRNA has to be processed. And when we say RNA processing, what does that mean? Introns come out, 5' cap added, poly A tail added, then it's going to leave the nucleus, go to the cytoplasm where the ribosomes will come translate the mRNA strand. So all of this takes a while. Like for a eukaryotic cell, it might take about an hour to go from DNA to actual protein, whereas in prokaryotes, it's happening immediately. As soon as the mRNA is being made, the ribosomes attach to it and start making the protein, okay? So there's a time and physical separation difference between prokaryotes and eukaryotes, because eukaryotes have a nucleus. They're separated by time and they're physically separated, transcription and translation. Now, the last topic is mutations. I said that a little bit weird. The last topic, because I'm a little bit tired. The last topic is mutations, okay? Let me move my head out of the way. I'm going to move it up here. Or maybe I should just, I'll move it down here. Okay, that's probably a little bit better. All right, point mutations. Point mutations are just basically single base changes. So here is our mRNA, okay, and there are 1, 2, 3, 4, 5 codons. There's 5 codons on the mRNA, okay? And this is the wild type, meaning this is the normal mRNA sequence, and this is the normal polypeptide chain or protein chain, methionine, lysine, phenylalanine, and glycine, okay? So you have 4 amino acids because the last codon is a stop codon. Let's talk about the first type of point mutation. The first type of point mutation is called a silent mutation. The reason why it's called silent is because there is a change in the codon, but because okay, so this is the codon GGU instead of GGC, okay? So it's supposed to be GGC, but it's now GGU. So the mutation, GGC, the mutation is GGU. That third letter of the codon is changed. Remember, you guys, we said that the third letter is that wobble. That third letter, because there's a mutation in that third letter, it's most likely going to code for the same amino acid. GGC, GGU, they code for the exact same amino acid, glycine. So there's no change. There's no change in the protein. That's why we call it a silent mutation, because it's not a mutation that one can detect, because that amino acid is not being changed, and there is no change in the protein, okay? Next type of mutation is called missense. Missense, okay? This is where there is a change in the amino acid. It's going from GGC, okay? That codon is original is GGC, but now it's AGC. AGC. So the first letter of the codon is changed, and that's going to go from glycine to serine or SER, okay? So when you look at the when you look at the genetic code chart, AGC is going to give you serine or SER. This is called missense. Missense. Think of missense as being like mistake. This is a mistake, a mutation where you can actually see the mistake, okay? All right. The third type of point mutation is called nonsense, okay? Where there's going to be a change in the codon. So we're talking about this codon, which is supposed to be AAG, okay? AAG codon codes for lysine, LYS, but there's a mutation. It's now UAG, okay? So that first letter is mutated. It went from A to U, and UAG is a stop codon. So, once that ribosome gets to the stop codon, that's it. Protein is made, polypeptide chain is made. And you know what? It's not even a polypeptide chain, because all you have is just the first amino acid met, and that's it. The ribosome is done. It's a signal for it to stop making the amino acid chain, polypeptide chain. So it stops there. That's why it's called nonsense, because it stops early. It stops early before the actual stop codon is reached. One way to remember this is like, stop the nonsense. Have you ever had a teacher or a parent yell at you, stop that nonsense? Okay? So the nonsense mutation causes the codon to become a stop codon, and then it stops, protein is stopped prematurely. Okay? The protein is not going to be long, it's going to be short, it's going to be stopped prematurely. When do mutations affect the next generation? Okay, you know, when a mutation occurs in the DNA strand, like maybe in an egg or a sperm cell, it's going to be, you know, it's going to affect the next generation, meaning it's going to affect the offspring, right? Because if the mistake is in the egg or in the sperm, it can't be changed, right? However, some of these mutations are happening, like could be happening right now in our cells. When DNA replicates. When DNA replicates, it can change the mRNA, which can change the codons, which is going to change the amino acid sequence. That's not going to affect our kids. It's going to just affect our own bodies. It's only going to affect our offspring when it's a mutation in the egg or the sperm. Okay. So sickle cell anemia is a type of genetic disease or genetic disorder that's caused by a point mutation. So on the wild type hemoglobin DNA, there's 3 sequences here, CTT, but it's someone with sickle cell anemia. Instead of CTT, it's CAT. As a result, the codon, so this is our codon here, it's supposed to be GAA, which codes for an amino acid GLU. So GLU is the amino acid. That's the correct amino acid. Whereas when you look at this codon on the genetic code chart, GUA, that codes for a different amino acid, val or valine, okay, amino acid. And so GLU is actually a hydrophobic, let me just double check. No, hydrophilic, okay? It's I'm just trying to remember which one's hydrophilic, hydrophobic. Yeah, GLU is a hydrophilic amino acid, whereas valine is a hydrophobic amino acid. I'll have to double check, but I'm pretty sure valine is hydrophobic. And so if you have a change in the property of the amino acid, it's going to change the shape of the amino acid. Do you guys remember we talked about like the secondary, tertiary, quaternary structure of protein? You guys, the characteristic change in these amino acids from going from hydrophilic to hydrophobic, that's going to change the way that the R groups interact with each other in the tertiary and quaternary structure of protein. And so as a result, the whole protein structure is changed. Okay? We're going from hydrophilic to hydrophobic. This is an example of missense, where there's a mistake. The amino acid is changed. Okay? All right, and then there's another type of mutation, that was point mutation. Now we're talking about frameshift mutation. This is where the codon reading frame, the 3 letters, is completely changed, and it's going to affect everything after it, okay? This can be due to insertions or deletions. These are the 2 that are going to lead to a frameshift mutation. So these 2 kinds of mutations are going to cause a frameshift mutation. Again, this is the wild type, the normal amino acid sequence, met, lysine, phenylalanine, glycine. This is the wild type codon sequence. But let's say there's a deletion, okay? So instead of UUU, this third, this is the codon, UUU, which codes for phenylalanine, that U is deleted. So then when the ribosome is reading that mRNA, UUG is the next codon. UUG is that next codon because UUU is now gone. So see, it's called frameshift because like now all the codons move over, and now every 3 that is read as a codon is completely changed. Somebody in our class mentioned how like this could be like if you skip a question on your test, right, on the Scantron, everything after it is completely messed up, right? It's sort of like that. If you delete a letter from the codon, then everything moves over and everything is changed. Now we have UUG, which is codes for leucine, and now we have GCU, which codes for alanine, and everything afterwards is changed, so forth, okay? So that's deletion. And then insertion, where you insert an extra letter into the codon, that can completely change everything after. In this example, UAA is the codon, that extra U going in there, it causes it to stop prematurely. It's a stop codon. You can have deletions of more than one nucleotide. You can have insertion or deletions of 3 nucleotides at the same time. Okay, so here you have AAG missing. So instead of methionine lysine, that lysine is now missing, and then phenylalanine and glycine follows after. An example of frameshift mutations. So Filmer was asking, where would this mutation cause the most change, beginning or end? So if we have a mutation at the beginning or at the end, which one would have the most effects or the biggest effect or the most change? It would be at the beginning, right? If you had it at the end, it wouldn't make, it would make a difference, but at least the frame shift is happening later on, right? So cystic fibrosis is an example of a genetic disorder that's caused by a frameshift mutation due to deletion, okay? This is the sequence of nucleotides, and with cystic fibrosis, these 3 nucleotides on the DNA are deleted, okay, are deleted. CTT is gone. As a result, this TGGTGTT follows that A and T. So everything shifts over, and it leads to a protein that regulates the amount of chlorine that's coming in and out of cells. It changes that protein, which is going to cause a lot of, for some reason, respiratory problems and also digestive problems. There's mucus buildup in the respiratory system and in the digestive system. What's the value of mutations, though? I mean, we talked about mutations as being like point mutations and frameshift mutations, which can have damaging effects. But at the end of the day, mutations is what causes change, right? And so it can be, in some ways, a beneficial thing, okay? We're going to be talking more about mutations when we go over evolution. By the way, that is a photograph of Charles Darwin, which I know some of you guys are fans of. Smurfs. Any questions? What color would a Smurf turn if he held his breath? Right? Because you know how when people turn blue when they don't get enough oxygen. So if a Smurf is not getting enough oxygen, what color would they turn? Okay, let's do some practice questions. Hopefully we have enough time here. I have to get going soon because my son has soccer. But anyways, a portion of the genetic code, UUU, codes for phenylalanine, GCC, alanine, AAA, lysine, CCC, proline. Assume the correct code places the amino acids phenylalanine, alanine, and lysine in a protein. So basically what this is saying is that we have a chain of amino acids where it looks like this, ALA-LYS. So it's asking, what's the DNA sequence? What's the DNA sequence that would give you, which of the following DNA sequences would substitute proline for alanine? Okay, so this is what it's supposed to be, but this is what you end up getting. Okay, so I'm going to put in proline, P-R-O, and then lysine. Okay. So phenylalanine, the normal one, actually, let's just look at the mutated one because we don't even need to look at the normal one. PHE is UUU. Proline, we said, is CCC. Lysine is AAA. So it's asking what would be the DNA that's complementary to that. So then the DNA is AAA. G, 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 T, T, T. That's the DNA, okay? And so therefore the answer is D, okay? Let me double check. Yep, the answer is D, okay? Next slide. What is the relationship among DNA, a gene, and a chromosome? What is the relationship among DNA, a gene, and a chromosome? So let's go ahead and read the answer choices. A chromosome contains hundreds of genes, which are composed of protein. That last part is not true, right? A chromosome contains hundreds of genes, which are composed of DNA. That can be an answer. Let's look at the other answer choices. A gene contains hundreds of chromosomes. Nope. A gene is composed of DNA, but there is no relationship to a chromosome. Genes are found on chromosomes, right? A gene contains hundreds of chromosomes. No. Okay, so the answer is B. A particular triplet of bases in the coding sequence of DNA, so DNA is AAA. The anticodon on the tRNA that binds the mRNA codon is, okay? So what it's saying is that the DNA is AAA. Therefore, what's the mRNA? mRNA is going to be UUU. But you guys, the question is asking what's the anticodon on the tRNA is, okay? So then the tRNA anticodon opposite to the codon is going to be AAA. So the answer is D. A part of an mRNA molecule with the following sequence is being read by a ribosome. 5' CCCG, ACG, that's the 3' end mRNA. The following activated transfer RNA molecules are available. 2 of them can correctly match the mRNA so that a dipeptide can form, okay? The dipeptide that will form will be, so if this, okay, is the mRNA, CCG, oops. So that's a codon, and then the next codon is ACG. It's asking, what is the amino acid sequence? Well, and they give you this information. They give you the anticodon and then the amino acid that tRNA brings, okay? So let's figure out what the anticodon is then. The anticodon is GGC and then UGC. So what's GGC? GGC is proline. So that's the first one. And then UGC is the second one, so threonine, okay? So the answer is proline and threonine, B. Okay, let me just check my answers. Yes, that is the correct answer. All right, you guys, this is a tRNA. This figure represents the tRNA that recognizes the binds a particular amino acid, in this instance, phenylalanine, okay? Which of the following triplets of bases on the mRNA strand codes for this amino acid, okay? This is kind of a tricky one because they kind of flip this tRNA. We've been seeing the tRNA like this, where this is the 5' end and this is the 3' end, and this is the way that we read the anticodon from 3 to 5, right? We've been reading the anticodon from 3 to 5, but this is showing from 5 to 3, okay? So this is 5 to, if this is 5 to 3, GAA, so this is 5 to 3, then the 3 to 5 is AAG. So it's asking you guys, what is the codon then to AAG? It's going to be UUC, so the answer is D, okay? How is a template strand for a particular gene determined? It is the DNA strand that runs from 5 to 3. It is a DNA strand that runs from 3 to 5. It depends on the orientation of DNA polymerase, whose position is determined by the particular sequence of nucleotides within the promoter. Well, I read that it's fast, okay? The answer is C, okay? Some of you guys were thinking, oh, it's B. But actually, when you look at the DNA strand, you know, it's anti-parallel. And it really depends on the orientation of the RNA polymerase. I mean, the RNA polymerase can be going in this direction, and then you know, this strand can be the template, or the RNA polymerase can be going in this direction, and then for this side would be the template. So do you guys see how actually both strands of DNA could be the template? It's just going in opposite direction. So it really depends on the orientation of the RNA polymerase, okay? They both go in, you know, where the two strands can both go in the 3 to 5' direction. A biologist inserts a gene from a human liver cell into the chromosome of a bacterium. The bacterium then transcribes this gene into mRNA and translates the mRNA into protein. The protein produced is useless. The biologist extracts the protein and mature mRNA that codes for it. When analyzed, you would expect which of the following results. Okay? So the thing is that it says here that the biologist inserts a gene from a human liver cell. And we talked about how genes in our genome has introns as well. Okay, so what this question is asking is setting you up. It's saying like when they inserted that gene into the bacteria, the mRNA and the protein that was made by the bacteria was completely useless. It wasn't able to do anything, okay, because that protein that was actually being made by the bacteria included amino acids that were coded for by those introns, So the protein was longer, the amino acid chain was longer because it included those intron codes, okay? And then also the mRNA was longer because it included those introns. So the answer is the protein and the mature mRNA are longer than in the human cells, because in the human cells, there's spliceosomes that are going to splice out those introns, okay? If a structure of a TV show is analogous to the structure of a gene, then the introns of a gene would be analogous to what? The opening theme music, the segments of the show, the commercials between segments of the show, the commercials between the shows, or the closing credits, okay? So what it's saying is like there's a TV show that is a gene. But what would be a good analogy to, you know, to the introns? You know how we said genes are cut up, right, into little pieces, but what's in between are the introns. So it's going to be the commercials that are in between the show, okay? Each of the following is a modification of the sentence, the cat ate the rat. So the cat ate the rat. Which of the following is analogous? This is like an analogy to a frameshift mutation. You guys remember there's two kinds of frameshift mutations. There's deletion and insertion. Those two cases is where the codon frames are completely shifted over, you know, because of a deletion or an insertion. So which of these? Okay, cat and rat are switched, okay? So that's not an example of frameshift mutation. Here's cat, rat, what is this? Okay, to me, this is like, it's all jumbled up, okay? Oh, I think this is like an inversion or something, okay? And then the cat, the rat. Oh, okay. So this is a, what is it called? A point mutation, right? A point mutation. The T became an R. The cat ate the rat. Okay, so this is a point mutation. Let's see. The cat, the rat. Okay, so there is a missing letter here. This is deletion. So this could be the answer. It could be D. And then over here, cat ate the rat. And actually, this is a deletion, but it's a deletion of 3. So it's not going to give you a frameshift mutation because things are not going to really move over and destroy all of the other codons. But this one is going to destroy the codons because it's missing a letter, and then everything over is going to move over. So the answer is D. Each of the following is a modification of the sentence. Okay, this is the same sentence. The cat ate the rat. Which of the following is analogous to a single substitution mutation? And we said that earlier, right over here with C, right? The cat, right over here, that T, it became an R. So the answer is C. Okay. All right, guys, we finished. So I'm going to go ahead and like edit this and I'll put it up as soon as possible, okay? All right.